Okay, so today I'm going to be unraveling Nemaline myopathy for you guys here. So let's begin with who I am. My name is Ishaan. I live in Palo Alto, California, and I'm a sophomore attending Palo Alto High School. I have interest in I have interest in the life sciences and the environment, which is why I decided to pursue this today, along with some other reasons, which I'll address at the end of my presentation. And my mentor was Sophie, who was super helpful through this entire project. And this was really my first experience with research, and she really guided me and helped me find good facts to support some arguments. And I wrote a review paper that I'll be talking about today. So this is what we have for the agenda, and I'll first give you guys an introduction of what Nemaline myopathy really is. Number one, the disease. Nemaline myopathy is a group of muscle disorders that primarily affect the body's mobility, and they vary in severity. These are diagnosed in early childhood or infancy, and they are marked by Nemaline bodies, which are rod or thread-like structures that appear in skeletal muscle cells with people with NM. So these are diagnosed by muscle biopsy. And NM is just short for Nemaline myopathy, and I'll probably be using it a lot in this presentation. Nemaline myopathy is prevalent in approximately 1 in 50,000 live births worldwide, although some isolated communities have much higher incidence with this dystrophy, like the Amish, where Nemaline myopathy is detected in an estimated 1 out of 500 deaths. NM, or Nemaline myopathy, is an inherited genetic disorder where there can be mutations in one of several genes. One of the main genes that I'll be talking about today is the NEB or NEB gene, and this accounts for about 50% of all Nemaline myopathy cases. Some other really important genes are the ACTA1 genes, and these genes play roles in the functional structures of skeletal muscles. While mutations lead to distinct muscle weakness in NM, NM is usually inherited in an autosomal recessive pattern. Notice how I said usually. We'll come back to that later. And this means that there are both copies of the gene have mutations. There are many forms of NM which can affect the patient's lifespan due to the severity of the disease. The neonatal form of Nemaline myopathy is the most severe form because infants who are born with this will have severe respiratory issues along with muscle weakness. Among individuals born with this form of NM, lifespan can range from a few months to a couple years, with most passing away during their early lives due to major respiratory complications or failure. So yes, it is a very rare disease, but I do believe that it needs to have some recognition and publicity, which is also part of the reason that I picked up this paper due to the severity, and it is a fatal disease. So yeah. With other milder forms of Nemaline myopathy, people can live through adulthood or even experience a typical lifespan, although still experiencing health problems and muscle weakness. Regardless of the severity or form of NM, most individuals with the disease experience some form of chronic muscle weakness, which weakens the muscle over time and can eventually lead to a disability, which may require different forms of motor assistance. Progressive weakness of respiratory muscles or chronic respiratory failure can dramatically change an individual with NM's quality of life, as they would need to be placed on ventilatory support. Let's move on to really deep dive into the genetics of Nemaline myopathy. Nemaline myopathy are caused by mutations in a variety of genes that play a role in the function of skeletal muscles where the issue is at its worst. So as I mentioned, the two most common genes are the NEB and the ACTA1 genes, but there are some other additional genes, a lot more rare, and they vary in severity, ACTA1 being the most severe type of gene, but the other genes are TMP2, TMP3, and TNNT1 and CFL2, which also can contribute. They are less common forms of the already rare disease. Although these genes can cause Nemaline myopathy, they're all very unique. The NEB gene provides instructions for making a protein called nebulin within skeletal muscles. Nebulin is found inside sarcomeres, which are necessary for the muscle to contract, which means that they tense up and become smaller and tighter. And muscle contraction is also a very common word into like the gym and athletics community, as people who work out and people who like lift weights. I'll give you an example, like the bench press. When you go down, when you like extend out down in the deepest form, that's when the muscle contracts, and that is just important as pushing back up the stretch. And those are how you build muscles. So people with Nemaline myopathy, they don't have the ability to do this, and you might not realize, but your muscles are always contracting your day-to-day lives. So muscle contraction is necessary for muscle growth, and when the muscle does not contract, the body starts to think that it does not need to use the muscle anymore, causing it to decrease in size and strength. In patients with Nemaline myopathy, the muscles do contract, but are often weak or inefficient, and they just get worse over time. Over 60 mutations in the NEB or NEB gene have been found that cause Nemaline myopathy. Some mutations insert or delete genetic material within the NEB gene, whilst others change a single amino acid, resulting in the lack of ability to produce the necessary nebulin. The ACTA1 gene, like the NEB gene or the NEB gene, provides a set of instructions to create alpha-actin or A-actin, which is an important protein for cell movement. A-actin is part of the actin family where ACTA1 plays a role in maintaining the structure of actin filaments, which are important for cell division, cytokinesis, muscle contractions, and more, meaning that a mutation in the ACTA1 gene can lead to severe forms of the Nemaline myopathy. The TMP2 and TMP3 genes encode tropomyosin isoforms that are also meant to stabilize actin filaments. The primary inheritance pattern of Nemaline myopathy, as I mentioned before, is autosomal recessive, and that means that both copies of the affected genes will have to carry mutations for the disease to be present. But also, like I mentioned before, this isn't the only form of the disease, and the autosomal recessive gene inheritance is more typical for mutations in the. Oh wait, sorry. In fact, ACTA1 cases of NM can exhibit autosomal dominant inheritance, but this is rare and can result in more severe cases of the disease. Now I'm going to be talking about one of the most interesting parts of my presentation. One interesting thing regarding the genetics of Nemaline myopathy is the super repeat, which I'll be showing you guys a figure with shortly, which is a structural unit found in some genes, but commonly NEB. In different species, nebulin contains super repeats, each with a similar domain composition that consists of smaller repeats called simple repeats, each containing an actin binding site. The number of super repeats called simple repeats the number of super repeats was shown to correlate with thin filament lengths. Super repeats are essential for regulating length of actin filaments in muscle fibers, but mutations within this region can disrupt the proper structure, causing defective muscle contraction ability. Now I'm going to be talking about the pathophysiology of Nemaline myopathy. So NM are defined by Nemaline bodies, which are rod-like structures that are found in the muscle fibers of affected individuals. They consist of sarcomeric proteins and actin, and the accumulation of these disrupt normal muscle fiber function. Nemaline bodies are formed by miscomposed or aggregated folded proteins, and this is the result of mutations in NEB or ACTA1 genes. The presence of abnormal proteins interrupts proper sarcomeric positioning and organization. This matters because sarcomeres are responsible for muscle contraction, which is disrupted in Nemaline myopathy. The difference in a healthy muscle cell is that sarcomeres are arranged optimally, which results to efficient muscle contraction. And like I mentioned before, this is really necessary for mobility. But in Nemaline myopathy, the organization of sarcomeres is disrupted by the Nemaline bodies, leading for the muscles to have an inability to contract. Researchers have conducted various tests and experiments on different animals to be able to understand Nemaline myopathy better, most notably being tests on mice. Studies have found that in mouse models, there are shorter, thin filament lengths and decreased force generation by muscle contraction. There are two main types of NM that have been studied in mouse models, the most common ones, ACTA1 and NEB. In NEB mouse models, research has shown that there aren't major reductions in mouse lifespans, and in nebulin deficient mice models, rods are not consistently shown. So these are not really optimal for human research. NEB mutations for mice can be characterized by both missense and nonsense mutations, and there have been other studies and tests for Nemaline myopathies in other animals like zebrafish. In ACTA1 zebrafish model, impaired swimming ability is present with disorganized muscle structure that is proven by the Nemaline bodies present. And in zebrafish, there are three types of Nemaline bodies. One reason why studying zebrafish for Nemaline myopathy is beneficial for research is because genetic manipulation comes easier in mammals. Some of the most studied models in mice are nebulin knockout mice and mice with specific nebulin mutations. Nebulin knockout mice completely lack nebulin expression ultimately, and this causes severe muscle weakness. Unfortunately, although these models have been crucial in studying nebulin and myopathy, their phenotype limits the usefulness for studying long-term disease progression. Recently, a compound heterozygous mouse model has been developed, and this more closely mimics the genetics of most patients with Nemaline myopathy. This is because this model resembles Nemaline bodies and muscle weakness of those similar to humans without early lethality. Now, I'm going to be talking about treatment for this dystrophy. The treatment options for Nemaline myopathies are limited because of how rare the disease is and the complexity of the genetics that come into play for it. As of today, unfortunately, there are no cures for Nemaline myopathy, but extensive research is being conducted into myopathies, especially rod body myopathy. Currently, treatments are used to help improve patients' quality of life and also to help with managing respiratory complications and skeletal muscle weakness. Most of these treatments involve physical therapy and, if needed, mechanical ventilation to support severe respiratory issues. And some patients with NM undergo surgery to fix skeletal deformities, which is similar for surgery for scoliosis. Recent studies in NM have been focusing on the molecular mechanisms of the disease with the goal of addressing the harmful genetics mutations and proteins that are causing Nemaline myopathies in individuals in the first place. Now I'm going to be talking a little bit about CRISPR-Cas9. I do know that we're running about time, but this is very important. So due to the recent boom in popularity of CRISPR-Cas9, gene therapy has gained more attention than ever before. Gene therapy, a technique that involves modifying an individual's genes or introducing repaired genes into a muscle cell affected with a certain disease, is a promising route to go for future treatment regarding Nemaline myopathy. Scientists have been conducting different research and exploring possibilities to deliver functional copies of genes like NEB or ACTA1 into muscle tissues, which could improve and possibly restore muscle function. But approaches like this are still in the early experimental stage and could take years to develop. Another area of treatment that is being experimented on regards to the use of ASOs, which my mentor Sophie introduced me with as her lab is working with these. These are short for antisense oligonucleotides, and ASOs are short RNA or DNA molecules that are used to change the expression of certain genes. They haven't been linked directly to Nemaline myopathy specifically, but around the area of congenital myopathy. And these could theoretically be used to fix splicing defects in NEB or ACTA1 and also moderate the production of proteins like labdulin and ACTA. ASOs have been experimented on in humanized versions of mice models. And yeah, here's some figures. So the top one I'll be explaining about a little bit. Nebulin, you can see it's the blue helix. It's essential for stabilizing the actin filaments. The CANK1 structure, I didn't really talk about this in my presentation, and its direct role to NM is less studied, but it does contribute to actin cytoskeleton stability, and that's what's disrupted Nemaline myopathy. Desmin is an intermediate filament protein that helps maintain the structural integrity of the muscle fibers. And abnormalities in desmin can further disrupt this muscle function. And the troponin and tropomyosonin, which are the brown and purple structures, are key regulators of muscle contraction. Yeah, that's what I have for you today. Thank you for having me on.
Oh my gosh, thank you, Ishaan. I have never heard of Nemaline myopathy before, so this was really like interesting, just totally brand new to me. I know you mentioned this in your presentation too, right? That it is, something with less attention, less recognition. So I'm curious how you learned about it and what sparked your interest in it.
Yeah, I mean, I've always really been interested in like muscles and athletics and stuff like that. And my mentor, Sophie, has been working about it. And I guess I kind of got interested in this topic first by my grandfather. He's had like a history of knee problems and he actually got knee displacement surgery, I think when he was 91.
Mm.
And things like the muscle cell, they really interest me. And my mentor, Sophie, who is also doing research in this, and both those experiences kind of prompted me to lead to Nemaline myopathy, which I've now learned is like a very severe disease and it does recognize more publicity, which is also one of the main reasons I decided to pursue this paper.
Yeah, I'm so glad you did. I'm like glad to know about it. Um And I love too that this kind of came from like a personal connection and interest with your grandfather, and then also to like your mentor's kind of expertise. I think that's kind of the perfect like blend to create a really cool Polygence project. So I love that.
Thank you so much for having me.